Thank you. So today I will give you a presentation about biocompatibility. And since the material that you will place in your body will come in close contact with the tissue, I will also give you an introduction to the world of biology, because that's where everything ends up. And if you have any questions, please feel free to interrupt me, or you can take it afterwards if you have any questions. So a short overview, I will start with an introduction, just giving you a short background about biocompatibility and what's happening during the history. A little bit about material properties per se, what we are looking for in materials. Then going into the world of biology, talking a little bit about immunology and biocompatibility, and finally I will try to summarize this in a short way. I think that if you look from a practical point of view, all of you will agree with me that our body is very good to have, especially if it's functions as it should. I mean, it's facilitated if it's possible to use your leg if you want to go somewhere. And in the same way, it's very good to have functional teeth for not only be able to give a beautiful smile, but also to be able to eat whatever you want, and also to speak without any problems. And this has made people to solve the problem if you lose a part of your body that you have to replace it. And already during that decent time, if people lost their teeth, they tried to replace it. For example, in Egypt, they used wood and gold wire to produce new teeth. As time passed by and we entered into the Middle Age, there was a great advantage. From the beginning, people used parts from animals as bones to replace the lost tooth. They were also tried to use teeth from corpse to put it in. This was not that popular because these teeth from the corpse actually was discolored and also could smell quite badly. So this was just for a short time. There were also some tests to transplant, taking teeth from one living person and put into another one. Unfortunately, not with that great success either. However, into the modern time, the development of new prosthetic materials have continued. And today, it's actually possible to more or less change or get new parts of all of your body. Especially the teeth is something else. You could have the hip and knee replacement. You can change your heart, your lungs. It's more or less only the brain that you can't change today, but that might come later on also. And if you look into the restoration of teeth in the dentistry field, for example, if you lose a tooth or it's damaged, you can't fix it, then you could have a new crown in alumina, zirconia based with porcelain on it, looking more or less exactly the natural teeth. Also, if you have to replace a hip, then you could also do it with the materials. So if you look at the fabrication process and the materials that's used today, especially within the dental field, there is a demand to have high geometrical precision of fully sintered individual structures. That means that if you lose a tooth, you will mill down this, take an impression, and then this full sintered prefabricated crown that come back should actually fit perfectly, and you will only do it once. And it's possible to do today if you use homogeneous blanks with isotropic shrinkage. And also pressed materials could be a good start for doing these new individual structures. Another issue if you're going to have a replacement is that you have to do it to a low production cost. If it's too expensive, no one can afford it. And one way to do it is to have a centralized production. And Nobel Biocare was actually the first company to have a fully centralized mass production units of mass production of individual fully sintered parts. And today we have a production line where the dentist and the dental technician using the CAD system in a computer to create a new crown that will, through a server, be sent to our production units. And within 24 hours, this individual part will be sent back to the dentist. or the dental technician that put on some veneers, and then you have the final crown that you could put on the last tooth. And if we look at our automatic ceramic crown production, we used to start milling the inner surface in gypsum. We press the powder on and mill the outer surface. And what we end up is a green body that it will be fully sintered to have the shrinkage and the dense product that will be veneered and put in the patient. So if you have solved the problem to have the high geometrical precision of your individual parts, you could produce it to a low cost. You also have to have the right material properties, of course. And mechanical properties is one issue, that you have to have, for example, the right strength, the durability of the materials. But also chemical stability is one issue. And again, I will take some examples from the dentistry, that's where I work on a daily basis. With zirconia, I think a lot of you know that we have this problem with the low temperature aging phenomenon, where the tetragonal face of the zirconia could be transformed into a monophasic layer instead. And what we could see is that if you have a fully sintered prefabricated crown inside here, that you put on the porcelain on top of it, this will actually protect the materials. So you do not see any degradation or transformation, I would say. While if you expose it to the humid environment, you will have a fast transformation occurring. And of course, you have the reliability in the materials with the defects that could appear. It's always that it's the weakest link that will tell you how reliable this product will be. And for example, if you have granules that are too hard, then during the production process, when you press it, it might not crash. And therefore, you will introduce the weak link in the product that actually might give the failure in the long run. So if you have a material that you will place inside your body, the hip, the knee, or if you have a heart valve or whatever you need to replace, you don't have to take the aesthetic part into consideration. However, if you're working within the dentistry, today, when people should have a new tooth, they expect it to look exactly like the other tooth in their own mouth. And this should be the right color. You should have the translucency, the opalescence, and also the fluorescence of a natural tooth. So it's a lot of different things that you have to take into consideration when you should produce this. And today, it's possible to do it. So what we produce is the inner core here, what we call a coping, a prefabricated crown. What the dental technician do is to put on a lot of different layers. And it's not just one or two, it's a lot of it. And in between each of these steps, you have to fire the product before you add the new layer. And finally, you will end up with a very beautiful tooth, but it will take time, and of course, it will cost. So, even if you have good materials, you have good processes, and you have a beautiful aesthetic, to have a fully successful rehabilitation, you also have to take biocompatibility into consideration. Because the material that you place in the body or on the body needs to be in fully harmonizing situation with the surrounding tissue. And this is the same if it's dental, if it's the hip or knee, or you place it on the skin. If you don't have a material that are accepted by the body, you will have an adverse affection. So, if you look on your body, all of you know that your body consists of a lot of different organs. You have the lungs, you have the blood, you have the heart, you have the skeleton. And all these organs are built up of cells. And what you can see here is the eukaryotic cells, and eukaryotic cells, that's our cells that we have. On the left side, you have the prokaryotic cells. That's the bacteria cells. So our cells will function together, building up organs, while the bacteria could function on its own. And there are some similarities, and there are also some differences between these two cell types. For example, the genetic materials of our cells is enclosed in the nucleus, so it's tight within one compartment. While in the bacterial cells, the genetic material will actually move around quite freely. It might be a little bit anchored to one of the cell walls. Another difference is the ribosomes. The ribosomes is our factories in the cells that produce our proteins. And proteins is something that we need to be able to build up new structures, for example. Both our cells and the bacterial cells have protein factories, but they differ a little bit. And another difference is around the bacterial cells, they have a cell wall. We don't have that one around our cells. And this is extremely important for the bacteria to have because this will protect it against intruders or the surrounding. We have other way to protect ourselves. So these differences that we could find between our cells and the bacterial cells is actually something that we are using. If you have a bacterial infection, you need a treatment with antibiotics. And what we do is develop antibiotics that only affect the bacterial cells, not our cells. So one way to do it is to go for the ribosomes, the protein factories. Since they differ, if we go for that one, only the bacterial cells will be affected of the antibiotic, not our own cells. And it's the same, since we don't have a cell wall, then we could make an antibiotic against the cell wall, and you will have an effect on the cell, or bacteria, but not our cells. But as I said, there are some similarities also between our cells and the bacterial cells. And what we have around our cell is not a cell wall, but we have a plasma membrane consisting of a bilayer of phospholipids. And this plasma membrane looks the same as the plasma membrane that the bacterial cells has, just inside their cell wall. That means that if we produce or develop an antibiotic against a plasma membrane, this will affect our cells also. And we have these kind of antibiotics, but you cannot take them orally. You just put it on your skin, for example, because if you take it inside, you will damage your own cells. As I said, the cell wall is extremely important for the bacteria because they do not have any other defense mechanism. We have that. We have the immune system. And the immune system consists of a lot of different parameters, and one of them is the white blood cells. And all blood cells in the body originate from one common stem cells that is located in our bone marrow. From these common stem cells, you will have a maturation in two different directions. You will have the lymphoid and you will have the myeloid maturation line. If we start with the lymphoid one, this will develop further on and mature into T cells. And T cells are the cells that's going around in our body looking for cells that are not our own. That could be if you have cells that's transformed into a cancer cell. They will be a little bit different than the cell normally are, and they will be attacked by the T cells and killed. It's the same if you have a transplantation, if you have new lungs, for example. Even if you got them from another human, they will look a little bit different, and they will also be attacked by the T cells. So that's why you have this immunosuppressing medication if you have a transplantation. From this line, you will also have the B cells. And the B cells could be further developed to plasma cells and memory cells. And these are the cells that produce our antibodies. So if you, for example, get an infection or if you have a vaccination, the plasma cells will start to produce a lot of antibodies. And these cells will not survive for your whole life, but instead you will have memory cells. So the next time you run into the same virus or bacteria that you have antibodies against, these cells will remember this and very rapidly turn into the plasma cells and start to produce antibodies. If you go for the other line, you have different cell types here. You have the mast cells. The mast cells are involved in allergic reactions. They have a lot of histamine inside that they can release, and you will have effects on your mucosa that will be swollen, and you can have edema and so on. You also have the monocytes The monocytes is located inside your blood, and they have a large round nucleus. When these cells go from the blood vessel into the tissue, they will mature further on to macrophages. And macrophages are cells that could live for a very long time out in the tissue. And they are called the large eater. And I will come back to that a little bit later. Then the third cell type is the polymorphonuclear leukocytes, daily called the neutrophil. And this is, compared to the macrophages, only a small eater. And as you could see, you don't have a round nucleus here. It's multilobulated. And this is something that the cell uses when it's going from the blood out to the tissue. This will facilitate this. And this cell do not survive for long out in the tissue. From this line, you also have another cell type that's not shown here. It's the erythrocytes, the red blood cells. They transport the oxygen around in our body, but they are not involved directly in the immune system. So, if we look a little bit closer to the monocytes and the neutrophils, they are called phagocytes. And this name is based on the fact that they could phagocytose. They can eat up an intruder, the prey that they bump into in the body when they try to defend ourselves. The monocytes, they will mature into the macrophages, and the macrophages are, as I said, large eater. That means that they could phagocytose, eat up a lot of bacteria, for example, if we have a bacterial infection, without getting killed. And they can survive for a very long time out in the tissue after it had matured. However, if you get an infection, it will take a time before you have the macrophages at the site of infection, because if the macrophages are not there, they have first migrate from the blood vessel out to the tissue. The monocyte need to be mature into the macrophages before it can start eat up the intruder. The neutrophils, on the other hand, is very rapid. If you get an infection, within hours you will have a lot of neutrophils in the area. But they can only eat up a few bacteria, and they will only survive a few days after they have left the blood. If you look inside a neutrophils, you can see the nucleus. It's just one nucleus, even if it looked like that you have several of it on this image. But what you also have is a lot of small vesicles inside the cell. And within these small vesicles, the neutrophils store a lot of toxic substances that are used when they defend ourselves. So, what's happening? If you place a foreign material in your body, or if you got a stick in your finger, what's happening is that in the area where you got, for example, your stick in your finger, the cells will start to signaling to the surrounding that something have happened here. They will release substances called chemoattractants, or if you have bacteria that you got an infection, for example, around your teeth, they will also start to trigger signals sent by the cells in the surrounding. And all these signals will diffuse away from the infected area and eventually run into a blood vessels. And the blood vessels that they will run into is the smallest blood vessels that you have in your body, called the capillaries. That's located the longest distance from the heart. They are very thin vessels, and in the wall you will only have one layer of cells sticking together. Inside here you will have neutrophils rolling around along the wall, or they are freely circulating inside the blood. When these substances that the cells have sent out bump into these, they will bind to receptors on the blood vessel wall. This will lead to signals inside these cells, and new structures will appear or disappear on the inside. And this will make the neutrophils that normally circulates or rolling around, they will bind tight and adhere to the wall and stay still, not move anymore. And the next step will be going from the blood vessel into the tissue. And this is a process called diapedesis. And here the cell have this lobulated nucleus that will facilitate the squeezing in between the cells to get out in the tissue. Once in the tissue, the cell can actually, through a chemical gradient, feel in which direction it should move through a process called chemotaxis. It's actually crawling. If you have seen amoebas, sometimes they're moving the same way. So they crawl along around in your tissue until it bumps into the infected area, where it starts eating up the intruders. And the intruders will end up inside the cells, where the neutrophils will try to kill them and destroy them. And this killing could be done in two different ways. The first way is oxygen independent. And these substances are located in these small vesicles, as I showed you on the SEM image earlier on. And these small vesicles will fuse with the large vesicle where you have the bacteria or the prey that they have eaten up. And releasing all these toxic substances will affect the prey. So it will be killed and hopefully also degraded. The other way it could kill it is oxygen dependent. And with an electron transport chain, the NADPH oxidase, you will produce oxygen radicals. It's superoxide anion, you can have hydrogen peroxide, you could have further converted into hypochloric acids, very toxic substances that also will help the body kill an intruder. So, normally when you place materials in your body or on your body, what you would like to do is to have it accepted. And for example, if you place an implant, you would like it to look like this. A lot of bone formation very tightly to the implants. But sometimes this does not happen. You will have more like what's seen on this side. You do not have a bone formation, something else has happened. And what can happen is that the neutrophils come to the area, they go into this prey or the intruder as they see the implant as, and they will try to phagocytose it, but it's too large. So what happened? Yeah, instead of giving up, the neutrophils start to release all these toxic substances and produce a lot of oxygen radicals. That will be released to the surrounding, and this will actually cause the inflammatory reaction. So it's not the bacteria, they will cause an infection, for example, but it's a body that creates the inflammatory reaction as a response on this. So all of these will lead to the inflammatory reaction, and if you're unlucky, this will actually lead to a failure of an implant. So if you go for the biocompatibility, what are we aiming for? Yeah, it doesn't matter if you're going to place it in the bone or somewhere else. You want to have an implant interface that you could alter, so you could have different surface structures, the topographic, you could change the chemistry, you could try to alter the energy or charge, as well as the bulk material that the implant is made of. So what is biocompatibility, if you look at it? Yeah, what you could say is that in this specific situation where you have placed this material, it's accepted by the tissue. And why? Why are some material accepted but not other? Is it the structure? If you look at the structure, you can look at it at different levels. You can have it in macro, on micro levels, and also on a nanoscale. And if you look at the macro, for example, if you take in the dental situation, if you're going to place an implant, depending on the situation, you can choose to have an implant with parallel walls, like this one, or you could have a tapered shape if you feel that this would fit better for this patient. You can go down on micro or nanoscale to change structures or patterns of an implant. And what you see down here on this image is a surface on our implants that called tionite with a lot of small pores where the bone actually grow into. So you have anchorage and acceptance of these materials. And it's very well known today that depending on how a structure or pattern is, you will have different responses from different types of cells. So what about chemistry? Will these be of importance? Yeah, if you place a screw in stainless steel that's resistant to corrosion, if you place it in building a house, for example, you will still have ions that's released in the body. So if you place this one, it will change. Also, titanium, that's normally fully accepted, will release ions in your body. If you have an oxidized material instead, as zirconia, alumina, or titania, this is more stable and less ions will be released. So there will be a threshold value that will determine if the ions released will trigger a cascade reaction inside the body. Will it be enough signals triggered to be above the threshold value and perhaps create this inflammatory reaction that we do not want to have? Or will we keep below the threshold and do not trigger this reaction? What we have to keep in mind is that my threshold levels, values, will differ from each of you in here because all of us are individuals. That means that you could say that within this range you probably have it, but all of us will be individuals. And also, if I have a threshold value today, if I compare it with the threshold levels that I had 10 years ago and that I will have in 10 years, I will probably have a little bit difference because you alter all the time. And the situation, if you have had a quite heavy infection before, you might be more sensitive than you are. And what the way you place the materials. If you do it in a very nice way and do not damage a lot to the cells surrounding, you might not trigger the signals to get this inflammatory reaction. But if you have a bad situation, you do a lot of damage, you will trigger a lot of signals and perhaps you will also trigger this signal cascade leading to an inflammatory reaction and loss of an implant. So how can we look or investigate if a material are biocompatible or not? It's two main ways you can look at it. The first one is the in vitro test, and the other one is the in vivo test. So if we start with the in vitro tests, this is done with cells, cell cultures. And depending on what materials or what you would like to look at, you can use different cell lines. You have cell lines that comes from different immune cells, the neutrophils, or you could have cell lines that based on macrophages. You could have cell lines that mimic the bone or the cartilages or for heart cells. It's a lot of difference. And what you will investigate is the cytotoxicity, you will have the genotoxicity, you can look at how cell proliferation and cell differentiation will be affected of this. And everything will be done in a two-dimensional world. And you actually are not looking at the biocompatibility, more like a cytocompatibility tests. So this is a very good start if you have a new material to see what effect do it have. But you cannot draw too large conclusion. What you need to have is some complementary tests with in vivo system. And in vivo system means that you're tested it inside a living organism, inside the body. And this could be done in clinical situation, of course, with humans, but it could also be done in animal models. And depending on what material you're looking at, you can use different animal models. For example, you can have rabbits, dogs, sheep, or goat, or pigs. That's quite common to use if you're looking for bone implants. Other ways, you could use mice, you could use rats. There's a lot of different animal models, and depending on what you would like to look at, you will choose different models. And when you're working in vivo, you will have the total situation, the physiological environment in a three-dimensional world where you have a lot of different things that will affect it. And also, sometimes it's extremely important also to have the mechanical load to take into consideration, which you cannot have if you have just a cell culture. So here you will have a biocompatibility test. However, it's extremely important to keep in mind that if you do animal studies, Doesn't matter which one of the animal models that you use. You cannot just take the results that you have got from these models and transfer it into the clinical situation. Even though that, for example, with pigs and humans are extremely similar. Especially if you look at the organ levels. You could actually take organs from a pig and transplant it into a human, and it will actually be accepted more or less without much changes. So we are very similar in that way. The structures are a little bit different. There are some sugar structures on the cells that differ, but it's possible. But we do not do it because there is a risk of spreading diseases from the pig to the humans that we are afraid of. But still, even though that we are so similar, you cannot just take it from the animal model into the humans. So you have to continue with also clinical studies before you could say that these materials are biocompatible. So, trying to summarize this. What we are looking for when it is a biocompatible material is that you should have the right chemical composition, so you could avoid any adverse effect in the body. You should, of course, have excellent corrosion resistance in the physiological environment where you are going to place it. And, of course, acceptable strength and high resistance to wear. So what we are looking for all the time is to avoid a too heavily interaction with immune systems. The material needs to mimic the host to be fully accepted and successfully integrated in the body. So, thank you for your attention.